right at the tip of the ear are a group of cells that are very special. We call them stem cells. And just like the stem cells we have in our own bodies are involved in making our tissues and regenerating tissues, these stem cells are the special cells that are building all these structures. Basically, they're building the ear during development. Now, this is a side view. I want to take a slightly different view and look from above, and it looks like this. So now we can see close in. This is the population of stem cells, this little dome of cells. And in the background, you can see the structures it's making of these rows of organs that are going to make the seeds, the rows of seeds, as I said. Now, if we look a little bit closer, you can even see individual cells here. So these are the actual stem cells. And you can see little structures like here. They look like little hamburgers. These are actually cells that are dividing, right? So these stem cells are dividing. And when they divide, they look like this. And when they divide, they make extra cells, which then will make the organs. Now, if you wanted to ask, can we make corn that will make more seeds? Well, you know, how could you do that? Well, one way would be to ask, can we make more of these rows of seeds, right? We know we have this group of cells, and they're making these rows of organs that are making the seeds. Can we just add more of these rows early in development and make more seeds? Well, the problem is, you know, as you see, they're pretty tightly packed in here. There isn't much space to fit in any more of those rows. But what if we could just make this structure bigger? What if we could make more stem cells and make more rows of seeds? Now, that seems like a tall order to do, but it turns out that we actually know the genes that control the stem cells in plants. And this is work from my own lab here in Cold Spring Harbor, as well as work from many labs around the world who work on how stem cells are controlled in plants. And the way we discover genes in plants is by looking at mutants. So this is a mutant. This is a plant or an ear from a plant where one gene has been knocked out or one gene is broken. And you can see the effect of that mutation is the ear is much shorter and it looks kind of ugly. I don't think any farmer or consumer would want to eat this. But actually, if you look again early in development, it has a very interesting characteristic, which is you see that the stem cells now are massively proliferating. So the normal corn has this nice small organized group of stem cells. And this mutant, the stem cells are just growing out of control. This is kind of like a kind of cancer in plants where the mechanisms that would normally control these cells have completely broken down. Now, can we actually use this information? I mean, this is really interesting information to scientists who want to understand how plants grow, but can we really use this information to improve corn for agriculture? Well, it turns out that we think we can.